And the same goes for our next speaker, who we're really fortunate is joining us, and that's Dr. Romina Gazis. She is from University of Florida, and she is a plant pathologist with extensive experience in fungal biology and an interest in tropical plant diseases caused by other groups of plant pathogens. During her PhD at the University of Maryland and 2 postdoctoral fellowships, one at Clark University and one at University of Tennessee, her research areas were really diverse and included everything from fungal endophytes of rubber trees with the aim of finding potential biological control agents that could be used to, protect against trees, to the population genetics and genomic approaches to investigate the evolutionary dynamics and disease ecology of pathogen and vector systems like Laurel Wilt disease and also like thousand cankers disease and others. So, really happy to have you with us today because you can also give us a really unique perspective into what's going on in Florida, where you've been dealing with Laurel Wilt disease for longer, and you also have an agricultural crop that is really important that's being decimated by Laurel Wilt disease, something I care about a lot because I love avocado, but also really important, from the economic impact to your region. So thanks for joining us today.
Thank you. Thank you for the introduction. Thank you for inviting me. I am learning a lot about forest species. And so I'm really a forest pathologist by heart. I really love the forest, and I'm really concerned about this disease spreading and, killing more of the natives. So my name is Romina Gazis, and I'm an assistant professor of plant pathology at the Tropical Research and Education Center down in Homestead, Florida, just at the tip of Florida. And I'm also an extension specialist. So many of you guys have been working on the system for a long time, and I'm really grateful to be able to meet you through this Zoom, and just wanted to say thank you for your work, and I'm really leveraging on this work and it influences a lot my program. So you probably heard that or worked with Randy Plotz in the past. I am his replacement, pretty much. I took over this program a couple of years ago, and I'm going to present you some of the latest research we have been doing here. So it has been already mentioned on what is this disease, and that it has the causal agent, which is Rafaela lauricola, which is an Ophius stomatalis, and it's a vascular lethal pathogen, and its primary vector, which is the red bay ambrosia beetle. This was the original vector. These are some more images, so you can see how it looks in culture and what are kind of the symptoms that you can find in avocado, are very similar to the ones that you can see in sassafras, the staining and death of the vascular system, and some microscopic pictures of the fungus in culture. Now, it was mentioned before, species in the Lauraceae are the ones that are susceptible to this disease and to this pathogen. And one of them, unfortunately, is the avocado, which we all love. It has dominated the world, and we are all worried about how it's been decimating the industry here in Florida. One thing that is different, at least with the knowledge that we have now, although I think it will be similar when you start looking at other beetles in native forest, is that this pathogen is being moved by other ambrosia beetles besides the Xyloborus glabratus. Actually, this original vector is almost never found, and it was, as it was mentioned before, in avocado orchards. And instead, we are finding other beetles, other naturalized and native beetles that are carrying this fungus in their machengia. Now, it might not be the primary symbiont, so the amount is not a lot of CFU, so it's not the dominant fungus within this machengia. However, as you heard before, this pathogen is very virulent, so it really doesn't matter that you have thousands of CFU. As long as you have 100 CFU, that's enough to start an outbreak. And so it's really important to look at these alternative vectors. Here is the publication if you want to go and look and see which are these beetles that are moving the pathogen in South Florida. Now, because we have different beetles moving the pathogens, they have different behavior in terms of if they attack the lateral branches or if they go to the base of the tree. And here is a video of an avocado tree showing these beetle attacks. And as you can see, these are extensive beetle attacks that go all the way from the base to the top. So this is not very uncommon, unfortunately, to see in avocado groves. And you see these sugar volcanoes. So we do get these sawdust, beautiful frass noodles that you might see in sassafras, but we mainly get these kind of sugar volcanoes that are a reaction of the mechanical damage that these beetles cause when they attack the tree. And when you peel off the bark, you could see the entrance holes, and then you see the staining caused by the death of the vascular system and the fungus growing in this vascular system. So even though we only need one attack to introduce the fungus into the host and perhaps to cause death, we usually see a massive attack in avocado groves. Now, this slide is showing how the outbreak happens in an avocado orchard. Very different than when we talk about a natural forest, right? That you have a combination of hosts, and there might be clusters of the susceptible host, so you don't have a massive outbreak like we have in avocado orchards. Now, the average age of the avocado orchard locally is maybe 15 years, 20 years. So these are already big trees that are very likely connected underneath the ground. And they're very likely to be connected, especially because in Florida, we almost have no soil, right? It's very thin, very shallow. So roots to get nutrients really go laterally. So it's very common to see trees being connected. And also they are planted very close, just to have more trees in an acre. So what happens is you have a first attack by the beetle or any of the alternative beetles, and it inoculates the tree with the fungus. And as we heard before, it seems like there is one attack, and then there is a couple of days of repellents, and then there's more beetles coming, getting more fungus into the host. Now, the beetle is gonna build a gallery, it's gonna farm the fungus is gonna multiply in these galleries, and it's gonna systemically infect the tree. Now, it doesn't happen quickly, this spread of the fungus systemically. However, it does spread to the root system fairly quick. So we have developed some experiments, and we found out that the fungus can be found in the roots 14 days after the first symptoms appear, which is 10 days after inoculation. So in less than a month, you will find the fungus in the root system. And by then, it already went to the adjacent trees. So that means that in 6 months, you can lose more than 100 trees in a grove. So it's pretty bad. It's really, really bad. And I'm gonna show some more slides of this root graft transmission, which is one of the biggest problems that we have here, because you might be doing spot treatment on symptomatic trees, but there's multiple adjacent trees that will remain looking healthy until the population of the fungus within the host reaches the threshold of symptom development. So very sad views when you drive around Homestead. Just to let you know, Miami-Dade County, which is where Homestead is located, is the avocado producing region or county in Florida. So this is where most of the groves are located. And it is very common sight to drive around and see these dead avocado trees, or you will see abandoned groves, or you will see groves that have been stamped and cut down, given, they will sell the land for buildings, or they were just changing crops. Now, in some cases, you will see growers removing the trees, but then leaving dead trees on the ground, which is really bad because, as I explained before, there is root graft contamination, and you are leaving source of inoculum in the growth. Now, it has also impacted avocados in the urban environment. Here, people love to have avocados in their backyards, and we also have avocados along the streets. And so, you know, it is also often that you see dying trees when you drive around, and usually homeowners hope that the tree is gonna get better, or they just cut a limb. And then I'm always concerned about the impact of these urban trees in the epidemics, right? Because I have seen trees starting to show symptoms across a grove, and then I keep driving around the same street, and then I can see how the disease, the beetles in this case, right, are just crossing the street. Miami-Dade County is really an urban agricultural environment. So you have these orchards really intermixed with urban development. It also has impacted germplasm collections. At the Tropical Research and Education Center, we have been losing many of important genotypes, and that's pretty sad. There's also the USDA germplasm collection that has been affected. I know that they have moved their germplasm to Hawaii. So, you know, we will have to be taking steps to protect these trees. But as we speak, we are losing a lot of representatives that we had planted here at the station. So that's another threat that we are suffering from this disease. And as it has already been discussed, this disease is really spreading. In Florida, it's present in every county. So really for us, eradication is not a viable option anymore. We have multiple hosts, natural hosts. We have the industry being affected, multiple groves being infected. We have multiple vectors. So really for us is trying to live with the disease, unfortunately. And this is not a matter of if, but when it gets to larger avocado production areas. Florida only represents 10% of the avocado production in the country. Most of the production is in California with the Hass cultivar. We have the tropical avocado type of avocados. And really for us has been a challenge to call for resources to bigger industries. Tropical avocado is really not that valuable in comparison to Hass in terms of acreage and the value of each fruit. However, we have been really having difficulties on telling them that they should be aware of this disease, that it's gonna get there. It will get there. It might not be, the behavior of the outbreak might not be the same because we have different conditions. However, all cultivars of avocado are susceptible, including Hass and Lamb Hass and all these Guatemalan Mexican hybrids. So it will become an issue for them. And so besides the avocado industry, we also have to worry about all the native Lauraceae that are in Central America, Mexico, and South America. So even though we have not that many Lauraceae in the US, there is a lot more in the neotropics. So this is a continuous worry for us. So my lab is working in two kind of broad directions. One is a short to medium term objectives, and we want to improve the early and rapid detection techniques. We want to explore sustainable management practices, and the medium to long term objectives are more of in tune with was mentioned before about finding tolerance or resistance out there to be able to regenerate these forests and to give the industry germplasm that is tolerant or resistant to the disease. These are members of my lab. Not all of us work in avocados. I have other programs in my lab. However, we all put some hours into this project. We all love avocado. We are all worried about it. So we all contribute to this advancing research. This is the Avocado Laurel Wilt team in my lab. We have several projects going on, sponsored by different entities, federal, state, and local. And we are exploring different sides of the disease. Some of them are more in detail, like we are doing transcriptomics. We are trying to find effectors to understand the mechanisms of the disease, and maybe some management solutions that are long-term. And we are also doing very practical and applicable research, like the use testing some fungicides. One of the, I would say the first grant that I got was from the Forest Service, and I was lucky enough to be able to get this funding, and I'm grateful to them. And I'm using avocado as a model system because a lot of the knowledge that I'm gathering is really transferable to forest species. So I'm hoping to be able to convey that message to you. So laurel wilt management is complicated for sure. Rafaela lauricola is a highly virulent fungus. Low inoculum can induce disease. External symptoms appear after the vascular system is already compromised and the pathogen has already extensively spread, which difficults early detection, right? Once you see the symptoms, the fungus is already in the roots. Once you see obvious symptoms, it's already on the roots, and it likely has gone to the adjacent trees. There's multiple susceptible hosts, including wild and native species. So there's constant new introductions into plantings. That's very difficult for us, right? Because you might be able to contain an outbreak in the corner of a grove, but if this grove is located next to a natural area, then you will constantly have these beetles coming in and creating new outbreaks. So then you have the grower really frustrated trying to deal with this disease. There's two transmission modes, insect and root to root. So we need multiple management strategies. And there's multiple efficient vectors and a broad host range. So that allows for rapid pathogen establishment in commercial area. So, but I think one of the most challenging things is the difficulty that it takes to study this disease, right? Because we are dealing with a hardwood, we are dealing with a vascular disease. So there's a lot of factors that will influence the outcome of the host pathogen interaction. So we have age, we have cultivar, graft combination. In avocado, we use a rootstock and we graft a scion. The scion is the commercial variety that we want to harvest the fruit from. And sometimes growers have in their grove even 3 grafts. So then you have a multiple cultivar in one tree, and they will react differently to the fungus. They're all susceptible, but we have found that there's differences in disease progress. So they will eventually die. However, the time to death varies, and the disease progress varies among cultivars. So, you know, because there's so many variables, we need to use multiple trees, many trees, in order to get significant results in our experiments. These are some of the challenges. Having growers donate field trees is complicated and a challenge, of course, and I completely understand. If, you know, inoculating field trees in their commercial operation is something that they might have a problem with. Also, avocado takes a long time to grow. For instance, this plant has 3 years old. So for us to get a decent diameter size, you know, we have to wait for 3 years, especially if we are using grafted material. And of course, having big plants requires space. And then we are in South Florida, where you get everything from, you know, pests and other diseases. So really taking care of these plants through time is a challenge. Also, the variability on the rootstock, since South Florida uses seedling material as a rootstock, and there's very much there's a lot of differences in seed and how these seedlings will develop. Also, this is a vascular disease, so we need to have the temperature right for the disease to be homogeneous across the trees, and I have learned that the hard way. And greenhouse with AC, it's a commodity in South Florida, and I'm guessing in other institutions as well. So I have to do these experiments by batch, right? I can only feed 30 plants at the time, depending on the size. So that is also a challenge. However, we have tried different strategies, and a lot of them, or many of them, are grower initiated. Growers are really involved in my program, and so we are working with them to test some of these strategies. I'm gonna go through a few of them, but there's more than the ones that I'm gonna be mentioning. So the first line of defense, of course, we think about fungicides, right? When we think about a crop. So this research started at Plot's lab. He retired in 2019, but before that, he worked a lot on these testing different active ingredients. And after all these steps that you start in vitro all the way to field, he decided, or he realized based on the results, that propiconazole and azoles are the ones that work the best. And so propiconazole was the active ingredient that was selected for field testing. Now, it is important to mention that this product and this active ingredient needs to be delivered to the active xylem, which is the main challenge in this kind of research when we are talking about field-grown hardwood, right? It has a very complicated vascular system, and delivering the active ingredient is a problem, since in vitro there is effect on the fungus. So it's a fungistatic. It stops its reproduction and multiplication in vitro. So that's the challenge that we are facing, how to deliver this active ingredient into the active xylem. And so ideally, you will have a formulation that can be applied to the tree's vascular system and that can be used in food crops. Now, you guys are gonna face a different challenge, which is working with natural environments. Applying fungicides is not ideal. But in the case of avocado, what we are facing is the challenge that it is a fruit-bearing crop, right? So we need to be careful on what we put in. The residue in the fruit that you're gonna eat cannot have any of these trace of this fungicide. And so, and ideally, we'll also have something that will remain on the tree, so you are not required to keep applying the formulation or the product into the system. And so Randy tested different products. Some of them are actually formulated for injections, and one of them is formulated as a spray. So just to let you know, Tilt is a spray foliar formulated product, and therefore it's oily. So it is designed to stick on the foliage. So that's the reason it might not be, and it's probably not, the best formulation to use as an injection, right? However, this is what we had, and it was proposed as an emergency label, and it was labeled under that. And actually now it has been permanently labeled. Avocado has been included in the label as for now, as an injection, to be used as an injection only. And so there was different type of injection that were tried. One was the macro infusion, which is not, it's not really an injection, but it's kind of like an IV type of system. And you will put these injection places at the base of the tree, at the root flares. And then the other option was the micro injection. So in one case, you use high volumes with diluted fungicide, and on the other case, you will use small volumes and you will apply pressure. And it really looks like an injection. I mean, it's really interesting to see. And there are some growers that are applying this management strategy. Now, the question is, does it work? right? So the active ingredient works. In vitro has shown that small concentration of this active ingredient stops the growth of the fungus. Now, it does not kill the fungus, it stops the growth. That's the mode of action that this active ingredient has. But more importantly, we have seen that it really, there is a lack of distribution of the product into the tree when injections are used. And you can see here in this slice of this trunk, this is where the injections were put in, and this is the growth of the fungus. So this bluish kind of stain is where we isolated the fungus from, and this is where the injection was done. So basically, the product does not move through the xylem, right? It's very unfortunate. And also it can cause damage to the vascular system. And there is some anecdotal evidence that that damage can even call for more beetles. And so, it's very challenging, the delivery. Now, there are some growers that are using it. There's, quite significant number of acreage that is being injected. And the growers say that it's slowing down the spread of the disease. Now, besides that, even if that is true, it is not a sustainable solution, right? Because it is very labor intensive and very expensive to do these treatments. And so when Randy was doing, and his lab was doing this research on fungicides, they found limited efficacy of drenching fungicides. And I believe that was because of the poor state of the root system. So my lab is now having a little bit of hope, because we are testing the application of tilt. But remember, I know the tilt is not the best formulation. However, this is what we have. If we prove that it works, it is easier for the company to label for drench application, so growers can have a tool right now. We need to work on more sustainable long-term solutions, but we are trying to find a solution also for tomorrow, right? So ideally, you will apply a drench application, and we are having some good results. So this is a trial that we just finished using Toro Cañon. Toro Cañon is a Mexican variety, and we were lucky enough to have clonal material. So you will expect an homogeneous disease development using clonal material. These are 3-year-old plants, and we drench them with tilt at the injection rate level. And before that, we treated the roots with Ritomil, which cleans them from Phytophthora and other common root rot issues. We follow a very good irrigation regime. So we really took care of those roots. Now you can see the controls are toasted. I mean, really, really dead. And we only saw one plant which had the smallest diameter showing symptoms. Now, these are very promising results. We still have to open the trees and plate the trees, because asymptomatic avocado trees can harbor and be extensively colonized by the fungus. So we still have to know if it has stopped the spread, the multiplication, or just the symptom development. We have also done some pre-trials in the field, and here you have an avocado tree in a commercial orchard that had been drenched with the same rate of tilt and then inoculated with 1 million CFUs. Now, this is a way over the amount of CFU that you need to induce disease in a big tree. However, we want to be sure that we are, given the the highest CFU that we can, so we know that if it survives, then it can stand multiple inoculations. And you can see here that the branch that has been inoculated does show symptoms, so it does get the disease. However, so far the tree is still standing, and only the branch that was inoculated show wilt symptoms, and the disease has not spread to other limbs or to the adjacent tree. So that's good news, because even with symptom development, the grower can come and cut the limb, and then the grower will not lose the whole tree, right? And that's one of the things that we are trying to achieve. Now, another management strategy that growers have tried is the stump and bag treatment. Now, the idea of this is to stump the tree and bag it with a really thick plastic bag under the South Florida sun, which is pretty hot, and to pretty much solarize the stump, right, and kill the fungus. So the growers had a very good idea, and this is based on a publication that showed that the Rafaela lauricola stops growing at 32. And this temperature for sure is reached within the stump under this bag. And the idea was that you would, the idea started with first removing the sick tree, and then stamping the adjacent trees and bagging the adjacent trees. This is called stump and bag barrier method. However, as you can imagine, growers are very hesitant on stamping a tree that looks healthy, even if you tell them it's very likely that already has the fungus inside the vascular system. They, of course, don't want to lose the production of these healthy looking trees. And so it suddenly became a stamp and bag technique, where they will stamp the symptomatic tree and bag the tree. Now, this method might work to reduce and kill the beetle population inside that stump. However, it does not kill the fungus. And I'm going to show you some research that is showing that. And so at the beginning, growers were seeing all these different outcomes. In some cases, the stump will resprout with a healthy canopy. And this healthy canopy will remain for a couple of years, actually. In other cases, it will resprout, and in a few months, it will decline and look with Laurel Wilt symptoms. In some other cases, it will just never resprout. Now, I have sampled multiple trees in these different situations, and especially in the ones that look like healthy resprouts. And most, if not all of them, show the roots with the pathogen, like you see here. So what I'm trying to say is that a healthy looking resprout might still have the pathogen in the roots. And actually now I can tell you that these resprouts that we have resampled, they all have died from Laurel Wilt. So it's just a matter of time for them to collapse. More evidence, we put some plates with Rafaela lauricola in the shade house during the summer. So summer gets really, really hot. You see even 37 Celsius. So really hot. And we found out, like you can see here, the control, it's in the regular incubator, 25 Celsius. Then you have the AC greenhouse, so you see some growth of the pathogen. In the shade house, it can grow fine, because why? At night, temperature goes down, right? Also, roots under the ground have this buffer of temperature, right? So temperature really doesn't fluctuate that much underground. And in the greenhouse, indeed, the fungus stops growing. This is greenhouse without AC, so it gets really, really hot during the summer. Now, when we took these plates and put them back in the lab at 25 in the incubator, they grew again. So that means that the fungus is not being killed by the temperature. It just stops growing. More evidence. I inoculated some trees at the station, and I wanted to know if younger trees, which are younger tissue, which are these resprouts, right? The main trunk might have, in this case, these trees had 80 years old, these stumps. And the wreath sprouts only had 3-4 years. So really the stems are young tissue, right? And so I inoculated different quantities and different concentrations of the pathogen and found out that younger tissue needs higher concentrations of the pathogen to show disease. And you can see here the pictures. So that's another piece of evidence that this technique is not working. You are not seeing the symptoms because the quantity has not, the fungus has not really reached the population that it needs in order to induce tylosis and therefore branch dieback. This is just to show you that root transmission is very, very important, and leaving the stump in the field is very dangerous. This is an experimental plot here at Trek, and you can see how the disease has spread through the rows in which the trees were connected underneath the ground. Now, this plot is a 80-year plot. So back in the days, growers were not making trenches to put the trees. So that means that the roots were really superficial, so very likely to be connected. Now,
just a heads up that you have about just a few more minutes.
Okay, so just a little bit about tree removal. You can see they always say that a picture is more than 1,000 words. You can see here this grower is really removing their trees, and this grower is using the stump and injection methods. So you can clearly see that sanitation works. Now we have these kind of tough to see. These growers, what they do now is they pick the trees and they pile them in a corner of the grove. And then now we have this cheaper equipment, and it comes and really mulches down the trees. So hard to watch, but this is what is happening to the avocado industry. Now, there is no, not everything is bad news. As it was mentioned before, we are also looking for resistance or tolerance in the fields, and you know, scouting the commercial groves, and we have found few trees that are coming back to production. So this is very important, very different than what you guys are looking for. I'm looking for a tree that is tolerant to the point that it's gonna still be in production. And we have found that trees that collapse have this complete colonization of the vascular system laterally by the fungus. Instead, trees that are surviving seem to be able to compartmentalize the fungus, and the lateral colonization of the fungus is limited. So this is what we are targeting now. We don't know if there's a genetic component to this. And so we are now screening a lot of different germplasms. We are collaborating with multiple, with other institutions, and they are sending germplasm to our labs. We have developed this protocol that we can use very small plants, 3 to 6 months, which saves us a lot of resources. So this is what, you know, we are doing right now in the lab. And these are some of the examples. So you can see here, this bench has seedlings that have collapsed by the inoculation, and then you have others that remain healthy. Even some of them do show some symptoms, but they recover. So we don't know how this translates into field trees, and that's one of the challenges that we are facing. These are just a picture showing we have received hundreds of germplasm from the biggest aguacate collection in the world, which is located in Mexico. So there's some hope that some of these cultivars are going to be resistant or tolerant. One of the things that I really wanted to mention because of the audience is that I'm also very interested in forest species, and we gave a workshop few months ago to the Tennessee folks. And one of the things that I noticed is that trees are being stumped after they are confirmed with a disease. And when I go to the field and I saw the stumped trees, I was really worried, because this is what we are doing here, right? People are stumping trees and it's not working. So I asked can I peel this tree and can I peel the healthy looking tree around this tree? And sure enough, the trees were contaminated. So leaving the stump is not an option. It is not really good. It just, the inoculum source remains in the field. And another thing that I wanted to say is that you guys are dealing with a very complex scenario because you have mixed forests with mixed species. So really, when you can have the wrong impression that the canopy, the overall canopy of that patch of forest is doing fine, but really it's being masked by other species like oaks or other species creating this canopy and this effect that that patch of forest is healthy. And I would really encourage to look at other beetles because here we have 9 at least native and naturalized beetles that are carrying the fungus. So that's something that I would really advocate for. And with that, this is my email. UF has come with a lot of resources. We have extension publications, we have talks, we have a website for Laurel Wilt. I will be more than happy to give you that information. So you can just send me an email and I will be able to send you those links. Thank you.
Wonderful, and thank you for that great presentation. So much exciting work going on there. I mean, it's just really frustrating to think about the situation overall, but excited to see some of the kind of promising things you all are finding. And I see that there are some questions in the Q&A that maybe, I know that we have time allocated for questions in just a few minutes, but if you'd like to go ahead and answer any of those in the Q&A, you're welcome to while we get started with our next presenter. But again, just want to thank you for the work that you're doing and giving us that insight, because I think, as you mentioned, there's a lot that we can learn from what's been happening there in avocado, and kind of, we can make our own new mistakes, but at least we can try, to learn from what we know is not going to work, and try some new pathways forward. So thank you so much.